Welcome to NewYorkBIO's virtual breakfast series, a digital program started in 2020, bringing you fireside chats with leaders from across the healthcare spectrum. This week's episode features Karen Akinsanya, the Chief Biomedical Scientist at Schrodinger.
Good morning, everyone. Thank you for joining us. I know you're all sort of rolling in as we've opened the webinar up live. This morning, we have a very exciting guest, which I'll leave Derek to introduce you. But I just wanted to say thank you to Goodwin Procter for sponsoring our webinar series. Every week, I've given you a little bit of update. I feel like we're reaching steady state of the New York reopening updates. Every region will soon be approaching, if they're not, they're almost there, but phase 4. And the governor gave parents of New York students some good news yesterday in saying that schools would be allowed to reopen. So there was, I think, a collective sigh of relief, at least for a few minutes, knowing that our children might be our public school educated children might be going back in the classroom. Other than that, I know a lot of you are bringing your employees back to the office. Please let us know if you need any help or connections with sourcing things that you need, or reaching out to the state if you have questions about the processes that they have required for you. So with that, we're glad you're joining us, and I'm going to flip it over to Derek to introduce our esteemed guest this morning.
Okay, thank you so much, Jennifer. So it is our absolute pleasure to bring you Karen Akinsanya, who's the Chief Biomedical Scientist at Schrodinger, and who I've received at least 5 or 6 emails after we announced you as a guest saying that they were absolutely thrilled that we were going to have you on. So welcome, good morning, how are you?
Thank you very much for having me. I'm well, thank you, Derek.
That's excellent. So one of the things we like to start off a little bit here is with a little bit of an origin story, so people get a bit of an idea of how you got to where you got to. So do you want to give us a little bit of background?
Yeah, happy to do so. So as you can probably tell from my accent, I'm from the UK. I was born in the UK, did most of my education there, secondary school, university, postdocs, and my first pharma job, actually, I worked for Ferring Pharmaceuticals in the United Kingdom. I then moved with Ferring Pharmaceuticals to the west coast of the USA in San Diego, where I stayed for a while. PhD and postdoc, endocrine physiology and developmental genetics, quite diverse. And I think if you know me well, I've done a lot of different things that all, I think, lead up to what I'm doing now. After Ferring, I went to Merck. So I was at Merck for 12 and a half years here in New Jersey and in Pennsylvania. And finally, in my last role, my current role, joined Schrodinger in New York City, which is a sort of company that embraces technology, has created technology solutions that help us discover drugs. So I'm really delighted to be here and to share a bit more of my story and what we're doing.
Well, that's fantastic. As you know, as a bit of background, Karen and I first met. I was the head of business development at a small startup company that was spun out of MIT. Karen was the first person I met through Merck to talk about what our platform did, and I would say you're probably 90% responsible for making sure that transaction actually got to the people that it needed to get to. So you weren't even, it wasn't your area, and still you managed to help make the deal happen. So that's, other people have echoed similar sentiments. So you had brought up the fact that you kind of did a lot of different things, and I think a lot of that's really important. So you started in pharmacology at Merck and eventually got into business development and licensing. And can you talk a little bit about that transition from really either being at the bench or being hardcore science and onto the business development side of the equation?
Yeah, absolutely. So when I joined Merck, I actually joined in a clinical pharmacology role. So although most of my training has been in basic science, I was an academic in the UK. One of the things that I quickly realized at Ferring Pharmaceuticals was that when you are involved in the discovery of a drug, you don't often get to be the one that takes it into human trials. And fortunately for me at Merck, in my first role, I was able to run human clinical trials, first in human studies. And, that was a fantastic experience. I think it actually made me a better drug discoverer because I really understood what happens when you take that fantastic test molecule that you've co-invented and actually start to figure out what it does in people. That experience led me then back to the labs, back to the discovery labs. And at the time, there was a very important initiative to embrace external science, which you're referring to. Really, a lot of collaborations, a lot of interactions at the time, a lot with academics, but then increasingly with biotech. And I was fascinated by that. The idea that the lab was bigger than the walls of Merck, and essentially by collaborating, we could do so much more. So when my diverse background in both basic research, drug discovery, and clinical pharmacology led me to a role in business development under Roger Perlmutter's team, really looking at all of the science that was going on externally and doing due diligence, it seemed like a natural fit for all of what I'd done to date. And so I joined BD and learned a bit more about the business of science, which, as we know, is a really fascinating space.
Yeah, absolutely. So, and fast forward a little bit, and now you're at Schrodinger, right? So, Schrodinger, which just IPO'd, earlier this year, is a great New York City story, and it started out effectively as a software company, correct?
That's correct. Yes.
And go ahead.
Well, Schrodinger span out of Columbia University 30 years ago. In fact, this is Schrodinger's 30th anniversary. Span out of academia with the notion that you could disrupt molecule design by using computers. That was a very long time ago, and fast forward to 2020, and I think that has not only come to life, but is very active at Schrodinger and actually at a lot of different pharma companies and biotechs, people using the software.
So can you talk a little bit about your thinking when you went to join Schrodinger? So you're at Merck for a long time, and what were some of the things that really kind of drew you to the company?
Yeah, great question. So as we've already discussed, my career had until that point spanned discovery and business development, clinical development, and I think at the end of my tenure, I was in the sort of I had this opportunity to go to a lot of different departments at Merck, and that was a lot of fun. But I think when I reflected, what I really, really enjoyed doing was identifying those targets and figuring out how do we test these ideas, how do we test these therapeutic hypotheses in people, and the idea that, can we do that a little bit more efficiently? Could we test more of these ideas? Because as you know, the failure rate's very high in drug development, and the more ideas we can test, I think the faster we'll find those that actually translate into real therapeutic solutions for people. And so when I came across Schrodinger, I was at Merck, and we were actually using the software to solve a particular challenge in a program. It struck me that while one could use it on that specific idea, what if you could do this more broadly on a larger set of targets? And Rami Farid, the CEO, and I started talking, and the next thing you know, I'm at Schrodinger running an 80-person group doing drug discovery, but also trying to figure out the breadth of the target universe that we can apply this software to. So that's kind of how I ended up at Schrodinger, this desire to do more with the tools that we have.
So what is really kind of the best angle for Schrodinger software? What is the thing that it does the best?
Yeah, so maybe I'll just start by saying that, drug discovery, drug design has sort of continued to evolve all the way back from the 1800s. It's sort of been industrialized to the point now where we have very large teams of chemists and biologists working together to design molecules. The issue is again, if we go back to the question of, why do we fail often, and can we create these more improved, sort of better molecules that really are well positioned to test the hypothesis. Drug design is very complex. You've got potency, selectivity, you've got the question of drug-like properties, and the Schrodinger software is basically physics-based. It's atomistic level modeling of how molecules interact with each other. And so imagine a universe in which you can explore vast amounts of chemical space on the computer, not synthesizing the molecules in a sort of iterative, empirical way, which is mostly how we do it, but that you can ideate on the computer which molecules, which subset of molecules are going to bind with potency and affinity to the protein of interest, or which proteins are going to interact with each other that have, really strong interactions. And then if you can do that on the computer, that gives you an enormous amount of headroom to then design in all of the other properties that you need. And having this sort of resolution of crystal structures or cryo-EM structures that allows you to really understand structure function and understand how to design these molecules with precision. The physics-based software essentially allows you to do that in silico. So we actually use it to do druggability assessment right up front and to kind of figure out whether we're even going to be able to drug a target. So you can imagine the savings there in terms of, okay, this is going to be hard with a small molecule. What about a biologic? So you can do that, but then refining at the same time all of the properties that you need into these molecules, that can be done using these physics-based methods. And essentially the result is that you can do this very quickly. You can identify molecules within months. You can then refine those molecules and do this, what we call multi-parameter optimization, to dial in all of those properties you need for the development candidate.
And it's interesting if you think about it, in thinking about the way that you're encapsulating this, a lot of it has to do with the person behind the computer as much as the tool itself, right? Because there's, with the level of hype and interest around AI, there's a common perception, I think, that you feed a computer a bunch of data and congratulations, it spits out a magic bullet molecule, which is unlikely to be the case. And it's funny, Bruce Booth had a blog post a while back thinking about machine learning and AI. And he said, the first time this was supposed to change everything was structure-based drug design, right? And what you're hitting on is really the importance not only of the actual tools, but the team itself. I don't think we're ever really going to outrun biology or outrun the number of factors that need to go into a molecule or a drug just through an AI platform.
That's correct. I think the popularity of machine learning and AI in some domains is very appropriate. If you're doing image analysis, for example, there is a real advance in terms of image analysis, and we use it every day in our lives, right? But when it comes to designing a molecule and figuring out how that molecule interacts with another protein, or a small molecule interacts with a protein, these are physics-based interactions. And so while machine learning can help in terms of looking through existing data sets, which is one way it's used in drug discovery, it doesn't really help you when you're coming to the next protein that hasn't ever been drugged before, or that you don't have a chemical library for. And so when you go to these first principles methods, you can really make advances in understanding at the very fundamental level what these interactions are about. And as you said, our teams are made up of chemists, modelers, and biologists. We have this sort of multidisciplinary team that are basically understanding what these calculations are telling us from a pharmacology point of view, because ultimately when you've selected these molecules with the help of the computer, you do need to test them. And so that does require this multifunctional or cross-functional team. And we do apply AI and ML to some of what we do. It's just not the foundation of what we do.
It's also probably something where you can't necessarily apply it the same exact way every time, because once you get to a particular starting point with a molecule, the questions change, right? So AI and ML are very, very good at optimizing for certain things, but a lot of times it's dirtier than that, right? You don't know, if you optimize for one thing, the optimal in one parameter may actually be pretty bad with respect to other parameters. So it's that balancing game where you don't necessarily just need the right perfect answer. You need an answer that you're pretty sure is in the right direction around parameters that you can test.
That's exactly right. I think interpolation is something that machine learning is good at. I think when you're in new territory, really these physics-based methods are very powerful and allow you to move into the chemical space that you simply, I mean, if you think about it, we're often asked, how large is your compound library? And all the chemicals in the universe is thought to be about 10 to the 60. We don't claim to go all the way there because not a lot of those, well, a certain portion of that is not drug-like, but about 10 to the 40 is probably the size of chemical space. And we're routinely running billion compound, multi-billion compound screens, which is obviously something you couldn't capture in a sort of fixed library of compounds.
Yeah. Yeah. So it's interesting in thinking about in thinking about how to set some of this up. I tend to look at how the company is made up and where the company came from. And while I had known a little bit about Rami's background, I spent a little time with some of the other folks on Schrodinger's website, and the team is a very, very deep technical team. Right? It's interesting where you see that almost everyone has a very deep technology background, which is rare in a lot of ways. So was that one of the factors in terms of you thinking about this as a place where I could really go and thrive? Is there a lot of kind of like-mindedness around the way to approach science?
Yeah, it's actually a fantastic catch. Obviously, I came from Merck, which is an incredibly science-driven company, and my next home had to be a place where we start every conversation with science. As you pointed out, Rami is a chemist. He was a chemistry professor. He actually was part of the team that created some of the solutions that we now share with all of the industry. The head of science, Robert Abel, actually discovered WaterMap and is very involved in the drug discovery. In fact, I think our head of HR has a PhD as well, and we basically have an incredibly talented team of 200 or more PhDs who are working on the software, as well as this drug discovery team that's sort of embedded. It's a classic sort of pharma-trained, biotech-trained team. And our application scientists, the people who are inventing the software, it's a really highly technical group. Actually, it's funny for me coming from a pharma environment to a company where on one floor you have literally sort of coders who come from Google and other companies who one would more traditionally sort of think about with tech, who wanted to do something interesting with their code that can save lives and change lives. And so, on one floor, we've got this very tech-heavy group who was inventing the software, testing the software and making it ready for others to use. You have the drug discovery team. You also have, people who are helping others to learn how to use the software. But yeah, it's a really technical company. And what's really important is that the goal is not to stop here. We have these solutions that are great. When you think about the challenges ahead of us still in drug discovery, we only have these structures for a portion of the human genome. In fact, as an industry, we've only drugged a very small percentage of the human genome or the human proteome. There are about 20,000 proteins. I think at this point, the drugs that we have only address under 500 of those. We have a long way to go. And one of the important aspects is how do we get better structures of all of these proteins? There's a group working on that at Schrodinger. How do we do better crystal structure refinement? So that work continues. So there's an ongoing effort to discover and develop new solutions for drug discovery. And that extends to things like how do we figure out predictive models for other aspects of the drug discovery process that we don't really have right now. So yeah, it's a very science-heavy, research-heavy company that happens to have this software that allows us to also at the same time try and discover drugs either alone or in collaboration with others.
Karen, you mentioned the sort of the depth and breadth of your team. And one thing that was impressive when I was reading about you and Schrodinger in preparation for today is that not only do you have, sort of the top 20 pharma companies in your, your client base, you do drug discovery, as well as I think your platform is used in the data I saw was over 1,200 academic universities around the world. How do you figure out as a leadership team where your focus lies, and how do you divide it up across those important collaborations?
It's a very good question because fundamentally the software can be used to design molecules of all kinds. The history of the company is mostly around the life sciences and drug discovery. You're right to point out that if you pick up a magazine and you're looking at structure of a protein, that was probably something that someone used Schrodinger software to create. And so it's used very broadly, at least one portion of it is used very broadly for that. And academics, frankly, use it in their research to figure out, the beginning points of drug discovery. The question of focus is a good one because there is a material science division, which is fascinating to me. It's something that I'm not obviously directly involved in, but there's a lot of potential in the design of molecules for material science. I think the experience set, and this is something that Schrodinger focused on about 10 years ago, is you're not really good at developing this software unless you're using it. And so the focus there at the time was, let's focus on the application of the software. Let's not just hand it over to other people. Let's try and solve some big design challenges in drug discovery. And that was done with the creation and co-creation, I should say, of Nimbus. And then after that came Morphic and Relay and a few other companies that really leveraged this software and allowed us to solve these very big design challenges for some pretty challenging target classes. And through that learning, I think it became apparent that, this has really important utility in drug discovery. So that is a big area of focus for the company. Looking forward, I think we are very keen to focus on areas of biology where there is at least some evidence that there's potential therapeutic benefit, and to accelerate how we identify high quality molecules for those. The question you raised, though, about the other areas and the other domains of application of the software is something we're still working through. We have, I think, some really interesting and exciting days ahead in material science and in other areas of drug discovery. Biologics is something we're really interested in expanding the use of the software in. As you know, there are a lot of biologic and peptide molecules that are very successful medicines. And so that's an area that we will be moving into. And we have a very large research team that's constantly looking at these opportunities and figuring out, is this ready for prime time? Is this ready for the drug discovery team or collaborators to work with? And so, yeah, there's lots of opportunity, and we continue to try and focus our efforts and really deliver results on the pieces we choose to focus on.
You're clearly doing a very good job, because I also saw in your SEC filings that you retain 96% of your collaboration and your client base, which is a pretty good percentage by any marker.
Most people would take those numbers. Most people would take that.
Yeah, thanks. That's great. And I think as biotech has risen to be an important force, and obviously we're here talking with NewYorkBIO, but biotech all over the world is increasingly subscribing to the solutions as well. You said the top 20 pharma, but now biotech is this amazing growing force. And I think that that's also a space where we're seeing a lot of utilization of the software.
So is it fair to say that, that Schrodinger is basically pushing ahead? And is it fair to call you a drug discovery company, or is it fair to call you a biotech company as opposed to a software company now? Is that a fair characterization?
I think we are a biotech company in some regards, but the software is, as I've already described, a critical part of the company. And I think if you were to ask Rami this question, he would say, we're both. We are both. We are still developing these software solutions, still pushing the envelope on what can be done with computational chemistry in many domains, as Jennifer just said. But we do have this established drug discovery unit, which at 80 people now is like a biotech. It's sort of a piece that we think has a tremendous potential. But it doesn't necessarily define the whole company. I think we really have to think about this as, two strong pillars that will contribute to life science, but increasingly contribute to other domains as well.
one of the things you brought up a few minutes ago, I think is important to think about, because as you well know, now there is no cookie cutter way to get a drug developed and to market, right? You have basically every option under the sun when you think about either partnerships or when you think about external relationships. And the lines there are really blurred between how much do you have to do in-house and how much can you do with others. uh, you know, prior to you being at Schrodinger, you brought this up before. They had a foundational partnership with Agios. They were a potential they were a co-founder of Nimbus, of Morphic. So already they had the seeds of, being, pretty entrepreneurial and pretty innovative in terms of the way that they looked at partnerships, which I imagine dovetailed with your background as well, considering the number of methods that you looked at there. Can you talk a little bit about how maybe that kind of strategy has evolved within Schrodinger? Because I can't imagine that it's been a very narrow focus. I imagine you think of things in a pretty broad strategic continuum.
Yes, yes, fantastic question, and you have the history right there. I think back to this concept that a lab is not necessarily defined by the walls of the lab. We are actually a completely virtual company. Our lab extends around the world, and that's something that during this pandemic has been pretty clear, is that, when you have a global lab, you can actually generate data and keep your programs running, which has been really great. When you think about where science comes from and the ability for that science to impact patients' lives, it's impossible for any one institution to kind of ring fence that, if you like. And so the idea of collaboration, working with others to pursue these therapeutic hypotheses and discover medicines, is in the DNA of Schrodinger, as you said, with the companies that were co-founded. The philosophy, I think, still today is that, when you have fantastic founders and innovators who've come across some important insight from a biology perspective, and they want to go ahead and try and drug that insight, you can essentially do that through a number of different routes. One of the ways in which you can do that is to create new companies. And last year, a couple of new companies that are still in stealth mode were founded by Schrodinger with other important academic founding groups of scientists. So we do believe that there is still the need to collaborate with eminent and highly accomplished scientists who have these unique insights. We also think that we have an incredible opportunity to help in walking through this proteome, in identifying great targets internally, and in developing drugs ourselves. But we also think that there are capabilities that pharma has, that it makes sense for us to partner some of those programs for the clinical development and obviously commercialization piece. So I think when you look at the collaborative framework for Schrodinger, you're going to see all of those things. You're going to see, new newcos. Several of the companies that were co-created have either done IPOs or doing IPOs, are sort of established companies now that people know. Some, as I said, are still in stealth mode. And, we still have a lot of collaborations with pharma, even though people buy the software. They sort of want to use the work with the SWAT team, if you like, that sort of a sort of name for the people who actually create the software and who use it on a regular basis. So we have a lot of collaborations with pharma. So I think, again, you'll see all of those different modalities of collaboration continuing.
Do you work with any smaller companies? Do you take inbound collaborative opportunities as well as outbound?
Absolutely, yes, indeed. In fact, the inbound interest in collaborating with Schrodinger, even since I joined, I think it's traditionally been quite active. But even since I joined, it's continued. In fact, obviously this year, Schrodinger is a little bit more on the map, and there's a large amount of inbound interest. You may have seen the collaboration with Twist, for example. As I said earlier, we're really interested in, how one uses the software to design biologics. It's something, there's a platform or a product called Bioluminate. but there are new approaches and research approaches that the team are working on, and in collaboration with Twist, we're sort of pushing the frontiers forward on that. So absolutely, yes. Working with smaller companies, working with academic labs, that's still something that we do.
Mm-hmm. That's fantastic. So you among the larger ones, you've got some work with both Google and with AstraZeneca. And Google in particular, we saw the press release about your work with Google Cloud on COVID.
That's correct. Obviously very topical right now. You mentioned briefly AstraZeneca. That's another collaboration that spans both small molecules and biologics with Google. And actually it's a collection of pharma companies. So Novartis, Gilead, Takeda, which is really, Takeda sort of initiated this whole multi-company approach to how we address antivirals for COVID-19. So those companies, together with Schrodinger and Google Cloud, have come together to bring each company's unique capabilities together to see if we can identify antivirals for COVID-19 for obviously future use. Vaccines are going to be that first set of reagents that we use, but ultimately I think an antiviral is going to be important. And so collaboration with Google, actually, we as you well know, the software does require quite a lot of compute power, which is one of the reasons why it took a while for this to kind of really come into its own. We now have access to clusters of GPUs, and we work closely with Google Cloud on that for our own programs, for our collaborative programs, but now excitingly, we're using it in this philanthropic alliance to go after antivirals for COVID-19.
Yeah. I think everybody realizes that we're probably going to need an entire toolbox against COVID. And even the people that think about vaccines say we'll probably need multiple vaccines. And I think therapeutically, anyone that treats patients wants options, because there doesn't seem to be a simple magic bullet that's going to work for everyone at every stage.
That is correct. I think everyone sees it that way. And even if you have IV medicines to begin with, for those who are most ill and in hospital, ultimately oral drugs are going to be important, one imagines, for ambulatory individuals.
Yep. Mild symptoms, severe symptoms, hospitalized patients, at-home patients. There's a lot of different ways that this can impact things. Okay, Jennifer, you want to lob a question over?
Yeah, sure, Karen. We've had a question from the audience, and that reminds me, I think I forgot to remind everyone at the beginning to please submit your questions via the Q&A box at the bottom. It's rare that we have a guest on Tuesday that is New York focused, that we don't talk about patient foundations and advocacy groups, simply because there are so many that are headquartered in New York, and we have such a rich, I think, of course, the best collection of academic medical institutions as well as patient advocacy foundations, which provides—
I agree.
Good to agree. You would see Derek like move off screen. Which brings this really rich combination of opportunity, right, for drug discovery as well as patient groups for clinical trials. So we had a question from the audience, which is great, about how Schrodinger works with foundations or other drug discovery groups that are specific to a particular disease state. Do you work with them, and how do you find that they help with your drug discovery or your other clients that are using your platform?
Yes, it's a very good question. I agree. New York is the hub for lots of things, including patient foundations and an amazing array of academic centers. And so we have collaborations with, I would describe them as sort of alliances again. The Tri-TDI, I'm not sure if you're familiar with it. I'm sure you probably are. The Tri-TDI obviously brings together a number of academic institutions and sort of tries to find really breakthrough ideas coming from those institutions where drug discovery is required. And so we work with obviously individual institutions as well as these alliances. On the foundation side, the drug discovery piece at Schrodinger, the internal ideation of programs, is relatively new, but we try to focus on diseases where we believe there are really serious unmet medical needs, and where patients are actually teaching us about the disease. And while I can't point to a very specific foundation that we work with on the disease side right now, we are very aware of, for example, Michael J. Fox. We're doing work in the neurodegeneration area, and we interact with other foundations who essentially are doing an amazing job in identifying populations of patients who have particular phenotype and who teach us that there are certain drivers of disease. And so whether it's in New York, frankly, or it's a global foundation that really seeks to kind of help and partner with drug discoverers to ensure that we're focusing on the most important unmet needs, but also learning from the incredible work that's, for example, going on in genomics to understand the root causes of some of these diseases. These are areas that we are very active in and hope to leverage in order to work on the most important diseases for sometimes very small populations of people.
Yeah, thank you.
Yeah, this is something that's been echoed in a lot of our discussions where patients are really being brought much more integrally into the drug discovering process, and really kind of educating people on how things impact their lives. And when you think about specific phenotypes, you're now getting into what is this going to look like in the clinic? What should our trial look like? You realize that the drug discovery spectrum really turns into a relationship with the patients that you're trying to treat. Okay, we have a bit of a compound question here, because we have a question on both the types of competitors you see in the AI and ML space, and also a more granular question about kind of the amount of time and effort it takes to get to kind of first in human with your platform. So to kind of combine those things together, can you talk about the way that you guys do drug discovery and how it may relate to maybe some other approaches that are either computationally heavy or kind of a hybrid approach, and maybe try and quantify a little bit of, is it faster, is it better, or what kind of box would you put it in to kind of think about it from an advantage standpoint?
Okay, sure. So first of all, let's just take a moment to kind of think about how we do drug discovery, and then I'll sort of layer on the view of AI, ML, and companies working in that space. So as I mentioned earlier on, we require a crystal structure that gives you this atomic level insight into the protein and the pockets and the active sites of the protein. When we have that high quality crystal structure, the first thing we do is essentially dock molecules, whether they be natural ligands, peptides, any kind of molecule that docks into a binding site. We can start these computational assays, if you want to call them that, with these initial docking events. And once we find that we can actually reproduce wet data, so if you think about it, there are papers published that say the affinity of a molecule for the protein is X. If we can reproduce that in this computational assay, we're on the right track. We know that the computational assay is accurate, the crystal structure is accurate. From there, we then use that computational assay or that model to then essentially score compounds in silico, and we're talking billions of compounds sometimes, scoring them for this particular active site or this particular protein pocket. When you start a drug discovery program this way, what you often find is that the initial molecules you identify are either nanomolar or picomolar, and that means you have this incredibly potent hit, if you like. Most drug discovery programs will start with a screen or an AI ML model that finds the sort of closest thing, but not necessarily a perfect fit in the way that these physics-based methods do. And so I think the biggest difference is really the ability to get these accurate simulations of compound interactions. And the fact that you start with these incredibly potent molecules means that you're sort of 2 steps ahead. If you think about faster or better molecules, not only have you sped up that very first part of the process, you're often, and I can now say this, we have about 25 programs we're working on, and the ones that we run completely internally over the last couple of years, it's become very evident. You can be in the later stage of discovery, which is called lead optimization, in under a year in some cases, and even faster in other cases, depending on the protein. You know, some proteins are very complex. And that shaves off an enormous amount of time in the very front end of discovery. What we're finding is that then going through lead optimization to get to a development candidate, that can be done in around 2 years. So we have some examples that were faster than that. We have some examples that were slightly slower than that, but on average about 2 years to get to a molecule that is potent, selective, and has great drug-like properties. So we think it's both faster, for sure. I can say that. I've been in the industry 25 years, and being able to dial in all these properties in a couple of years for some pretty challenging targets sometimes is pretty incredible. And so, yes, it's faster, but then at the end of the day, when you're able to design a small molecule for something that was originally drugged as a biologic, that's quite an accomplishment, right? So coming up with a small molecule that really is not just potent, but has the properties that would enable it to be an oral drug, for example, that's pretty amazing. Or dialing in selectivity that has been challenging for decades. So we think both quality and speed are benefits of the platform. Now, when we talk about AI and ML, AI and ML, as we've described before, are very good at working with training sets, right? So you have a very large training set. As we know, when you're looking for pictures of cats, and this is the sort of famous YouTube example, if you feed the ML model lots of pictures of cats and maybe a couple of pictures of another animal, it will absolutely identify that cat, right? When you're working with a protein and you don't actually have the molecule in the training set that you actually need to either initiate or finish your drug discovery program, it's very hard for that model to actually find that image or that molecule in the data set that you're feeding it. If you're working on a related protein, of course, the ML model is going to be able to find related molecules. So there are utilities. We use it actually as part of our workflow because the physics-based methods are pretty computationally expensive. And using a combination of physics-based first and then ML to sort through the results of the physics-based methods, that workflow works really well. But it really does start with these accurate first principles methods. I hope that answers the question.
I think so. I think I think it comes close, right? It's difficult to boil the ocean, right? And I think what you've done is given us a really good picture of the way that Schrodinger approaches everything. And the question of will a tech-based drug company make things faster? In a lot of ways, they can make certain things faster, but ultimately, it seems like one of the best advantages is to be good at a thing and to have that thing lead to better molecules that go into the clinic and really strong and sound biological processes that you can test in humans, right? It's unlikely, at least to me, that somebody's going to revolutionize 8 different things at once and spit a drug out of a 3D printer that you can then take in your kitchen and ensure that it's going to be both safe and cure your disease. I think we're probably at least a year or so away from that.
Yeah, I've seen my kids 3D print, and we do not want.
Yeah, I think that's correct. There are certain parts of the drug discovery process that we don't pretend that we've disrupted, right? I mean, the safety assessment and all the things that have to happen up to an IND opening study, those are things that we don't claim to adjust. I mean, having a more selective molecule and understanding the off-target molecules, the off-target proteins, and being able to dial away the obvious side effects is one thing. But look, at the end of the day, it needs to be tested in vivo, and that's something we don't claim to be able to predict. And as you said, there are some amazing applications of ML and AI now in pathology and other aspects, even text mining to look for patterns in the literature. But these first principle methods, I think, is really about the foundation of drug discovery, in identifying the molecules and getting them into safety testing faster.
Karen, as you know, because you know the types of companies that we have in New York, we have a lot of earlier stage companies. And so we had a question. It says, could you please speak about the process leading to initiation of collaboration? And this comes from an early stage startup company.
I see. So we have an active business development group led by Connie DeCruz, and as I mentioned, inbound interest in terms of collaborations is something that we are very active in. The process really, I think, involves outreach with a very clear therapeutic idea. If it happens to be in the life sciences, I'm sure there are lots of material science companies in New York as well, but I can speak to the life science piece. If someone has a unique insight and they happen to have a crystal structure for a protein that they think really is going to enable us to identify novel medicines, that outreach can come in through our business development group. Some of it is appropriate for the drug discovery team to look at. Some of it isn't. Obviously, people shouldn't share compounds with us. We don't want to see any compound structures. Protein structures is okay. But yeah, it's really about just talking with our scientists and our business development group about the opportunity to collaborate and figure out if we can actually apply the technology. But your comment about focus is a very good one earlier, right? We can only concentrate on drug discovery for a certain number of targets and with a certain number of partners. So we will work through that process and see which opportunity is the right match.
Yeah, that makes sense.
Okay, I'd like to pivot slightly because I think, one of the areas that we genuinely would like to touch on is, the fact that you have, not only founded a not-for-profit in MyTech Learning, but also this goes all the way back to your days in graduate school and working with, with underprivileged kids in science. So I thought it'd be really good to actually talk about access to science and really how kids are exposed to science and really where our new scientists are coming from. So can you talk a little bit about, both your background in graduate school and how you got to put this not-for-profit together and what it does?
Yeah, thanks for the question. So one of the things that I've always been passionate about is the public understanding of science in one way, but it's become pretty apparent to me. I think I was a PhD student when I joined the Prince of Wales Trust in the UK and realized I was actually giving a lecture to a group of, I guess they were middle school students. And it became pretty obvious that there was children in that room who were clearly bright, who did not see themselves as having a path to being a scientist or a doctor or some other technical disciplines. They were, I think, under 12 and they were already making statements like this. And I was stunned. I mean, I had a very privileged upbringing. I had a microscope and a typewriter when I was 5. I had parents who could tell me what was going on down that microscope, both with training in healthcare and science. But it really struck me that there's a large number of kids who, frankly, probably have the inkling that this is an area that they would love to kind of explore and maybe have careers in, who unfortunately don't have access to what it takes ultimately. And so with the Prince of Wales Trust, I worked to try and mentor those kids. And all through my career, actually, I've found opportunities to work with kids or with, frankly, adults who are trying to make it in terms of careers in these technology, science, medicine fields. In New York, based in New York, the National Medical Fellowships is seeking to find ways to bring more diverse doctors into hospitals in the healthcare setting. And I was able to serve on their board for a while. So I'm very passionate about this topic of, how do we bring more science to kids at an early age before they give up on the idea that they might be scientists. And one of the things I always wanted to do was to be able to teach kids science myself. My daughter was fortunate to do a gifted and talented program, and she was learning things that were clearly very many years ahead of where her class was. And it kind of gave me the idea that what if we could do this for lots of kids? And my tech learning was something that I was working on with a few other folks. Really, the idea of creating a lab where kids could come and try and explore science experiments, because unfortunately, a lot of kids don't get access to the ability to run science experiments on a regular basis, don't get a chance perhaps to, as great as teachers and the education system strives to do so, giving kids more time with science, more time to explore and invent, I think is lacking in some way. And so we basically tried to create a space for kids to do these experiments, and it was an enormous amount of fun. In fact, I'm not actively involved in it right now for reasons that I think we're all experiencing. But one of the things that was very enlightening was a number of letters that I got from parents and texts saying, "Thank you for teaching my kids about immunity." I mean, some of these kids are 7 years old, and the fact that they understood what immunity was when they were that young, at some level, right? But especially now with everything that's going on, their parents recognized how valuable an experience what that was. And I would say that during these days of COVID-19 and just everyone talking about access to therapies, I think we're probably going to see a wave of kids who want to, create vaccines because they realize just how important that is. So yeah, it's something I'm really passionate about, and with all that's going on in terms of this social justice movement, trying to get equality, I think equality in terms of access to great education is something that I'm also very passionate about. If you plant a tree and it has good access to light, water, and good quality air, that tree is going to grow and it will turn into a forest. Any one of those resources that are lacking, it's very hard for that to happen. And so, think about our kids that way. They need access to great resources, great education, great mentors. And so that's something that I continue to be passionate about till today.
Well, that's fantastic, because really great minds can come from anywhere. And, very often you have, people that need the spark of curiosity and how to follow it. And, those sparks aren't necessarily evenly distributed. And having another outlet to try and do that is both fantastic and commendable. So, one of the things that we will do, I tend to post things on Twitter and LinkedIn after these conversations. I'll put up some stuff on the My Tech Learning link and a few other things with relation to that. But that's terrific. I mean, the next generation of scientists can come from anywhere. And like you said, I think COVID may give us a new wave of epidemiologists and virologists that come out. So
I know,
really the
my
vaccines that get developed in 20 years are going to be amazing.
Even my 14-year-old, I think it was in late March, he took an online epidemiology course from, I think it was UNC. And so, just to try, because he was hearing so much and reading so much, and then he starts his science research program for 9th grade. And so he is super excited about being able to go into a lab and do some, helpful, I mean, granted they're 14, 15, but it's just getting him excited. And I do think maybe that may be one slight silver lining of everyone talking so much, right, about this and around coronavirus and the drug discovery process that seems so public now, right? Whereas normally it wouldn't be as public, is that hopefully we will have, and you're right though, we have to provide the tools for that interest to blossom into actual scientists. And so thank you for the work that you're doing to support that.
Well, I want to shout out to Jenny Chambers, who's our head of education. She actually ran.
Oh, she's great.
Yeah, she ran a molecular modeling course for kids recently. I don't know if it's still up, but the exposure that kids get through her work, we work with a lot of foundations in New York actually that help to educate kids in terms of technology and software development, but extending that through to drug design with these online courses with Jenny, I think is a really exciting development as part of the lockdown and giving kids just something different to learn while they're all at home.
Speaking of online and lockdown and learning, I did notice on your website that you have the Schrodinger Summer of Science series, and that's available to everyone, right?
It is, yes. Schrodingers, publishes, I think, hundreds of papers a year on the science, but one of the other things that we're very active in is chemistry and molecular modeling and computational chemistry learning. And so we have kind of a Schrodinger university, but this year, with the inability to hold these sometimes live meetings where people come and learn, that's been shifted to this Summer of Science series, where we have a lot of our scientists, and I think some company scientists from outside, coming in and talking about how to use the tools, about the challenges in drug discovery. So yeah, it's a fun series that's up and running right now, and I think it is open to everyone. I think if you go to the website, there's a way to sign up.
If you turn your software into a video game, you're going to have armies of children that are working to design your next molecule. Right? You're going to have a great protein interaction that's going to be developed by a second grader in Utah.
I think there may be some projects like that. Our head of technology, Pat Lawton, I think, has been involved with some of the sort of 3D goggles, and there's another. It's called Looking Glass, and it allows you to kind of walk through proteins and really look at how compounds are docking into proteins. We don't obviously use that in the professional drug discovery group, but I think just that experience, back to what I was saying before of, running experiments and seeing what this is all about, I think could be an amazing experience for kids. And I think there are some large projects where large communities of people try to fold proteins and do other things. Again, not, I think, in the goal of necessarily discovering drugs, but it's great for kids to get immersed in this, I think.
Okay, we're winding down a little bit. So I would say I think we can kind of close on this topic. So if you could do one thing that would either, give more kids access to science, or if you could kind of add one thing to the countries of the globe's, lexicon or activities in and around science, what would you do?
I think if I had unlimited restrictions, I would say that just as we have coaches for running, and we have coaches for soccer and American football, or get this right. I think just as we have coaches for all of those activities, I would love to see in every town, in every city, science coaches working with kids, showing them what science is all about at the bench, and talking about how we as a global community can improve global health by focusing on science and technology, and just making it available at a local level for kids all over the globe.
That's the perfect.
I think that I think it's perfect. I think there are, I think if we recognize that science and creativity can come from anywhere, it would be great to stoke more of it. And, it's fabulous that you and honestly many of your colleagues are really working to not only think of those things, but breathe life into them and bring them out into the public domain. So, thank you so much for joining us this morning. This is wonderful. And we can't tell you how much we appreciate everything that you do, and for being our guest this morning. Thank you very much.
Thank you so much, Derek and Jennifer. It's great spending time with you.
All right, thank you so much. Enjoy your day, everyone.
Yeah.
Bye.
Thank you for tuning in to New York Bio's virtual breakfast series. Join us every Tuesday at 9 a.m. for more discussions with leaders from across the healthcare spectrum. For more information on New York Bio, please visit us at www.newyorkbio.org.